Okay, so we're now going to move on to Unify Pharmaceuticals. We've got Laura Dugan joining us, CEO. I hope Laura is with us.
Unify Pharma is a company that was formed a year ago to commercialize a novel class of anti-inflammatory small molecules that is the product of nearly 20 years worth of work. So even though the company is young, the technology has had a lot of investment. I wanted to tell you a little bit about the founders and the corporate management before I go into the science, just because I think that we have a strong management team, and it's been very exciting to work with them. So Professor Sir Mark Feldman, Lasker Award winner, is very well known for being the co-inventor of anti-TNF therapies. And as of 2012, anti-TNF therapies, including Remicade and Humira and so forth, are the single best-selling class of drugs in the world. And they are now being used for over 15 different clinical indications. So a very broadly important breakthrough in immunotherapies that open the door to many others of these. I am a physician scientist with a focus on neuroscience. I trained at MIT. I then was at Stanford, Washington University, UCSD, and now I'm at Vanderbilt as the Abram C. Schmerling Professor of Alzheimer's and Geriatric Medicine. I'm co-founder with Sir Mark of this. Our CEO and director is Professor Chazabuntra, who is at Oxford. He was the vice president and head of biology research at GSK, and he was involved in bringing nearly 40 different drugs to market during his period there. He is now the pro-vice chancellor of innovation at the University of Oxford. Our COO and CFO is Ozan Pamir. He actually has a background in venture capital and has actually been very successful in a lot of fundraising with that, and he joined us a year ago as well. Our CSO chemist is Dr. Chakraborty, who is a faculty member at Carnegie Mellon, a very capable chemist, and is overseeing our scale-up of chemistry of our compounds. And our SAB includes someone who needs no introduction, actually, Larry Steinman, who, of course, has successfully brought 3 companies to market with incredibly successful treatments. And then Dr. Dennis Choi, who was the executive vice president for neuroscience research at Merck for a number of years and is currently at New York Stony Brook as the just stepping down head of neurology there. And so we have, that is our corporate team. So the compounds that we are developing are quite unique chemically. They are catalytic small molecule, it's basically synthetic enzymes. And what our lead compound, UP1007, does is it catalyzes dismutation of 2 important biological reactive oxygen species, and those are superoxide and hydrogen peroxide, in the manner that the native enzyme does, and that is that the end products are oxygen and water. What it does biologically at the tissue level, however, is it's a very potent anti-inflammatory compound, and we have published over the last 15 years anti-inflammatory effects in a number of different organ systems, primarily in the nervous system, because that's my personal focus. The 3 first-line targets for UP1007 clinically include Parkinson's disease, and I'll present to you the PRIMATE data that is the compelling foundation for choosing that as our first-line therapeutic indication, but also acute macular degeneration, which actually has a strong inflammatory component and is only effectively treated in about 10% of individuals with the disease. And then mild cognitive impairment, which is emerging as a condition that may involve an inflammatory process, a chronic inflammatory process that might also be amenable to an anti-inflammatory intervention to slow or prevent progression to Alzheimer's disease. So the science behind the molecular mechanisms of UP1007 and the other compounds that are part of this panel of very unique small molecules has been reported by us, but also by several other groups. And so we have confirmation of a catalytic mechanism. It's a very high kinetic rate dismutation reaction. And the interesting thing is that we've also been able to characterize the biological compartmentalization of UP1007 and its other small molecule partners or compounds. And the UP1007 can access mitochondria. It actually distributes well into cells quite quickly. And it has a very nice biodistribution into both cytosol and into biological membranes. So inflammation is, of course, an emerging theme in not only diseases, but in diseases of aging and also the aging process itself. We have some compounds that are well known to be anti-inflammatory compounds, but they target specific areas of inflammation. And there's a third limb to the right there that involves innate immunity and the innate immune systems of oxidases, including the NADPH oxidase family of compounds or family of proteins. And this area of inflammation and innate immune activation is just not well targeted by current therapies. Anti-TNF therapies do have some activity upstream of this, but the issue is that anti-TNF does not, for example, get into, or antibodies at least, do not get into the nervous system. And so the brain is an organ that actually is not well targeted by most of these currently available anti-inflammatory drugs. So these are just a few of the many diseases and syndromes that are believed to involve inflammation. Neurodegenerative disease is, of course, where we focused most predominantly, but I'll mention that we actually have anti-inflammatory effects in disease models in lung, some in heart, and a little bit in eye as well. So the reason we have focused on the nervous system is because, as has come through the last 4 days, there's a great unmet need for therapies that will target CNS disorders. And we have a graph here that shows the increasing prevalence of all types of dementia as folks age. And we really have no good treatments for most of these. We have symptomatic treatments for Parkinson's disease, and we do have some preventative approaches to things like stroke and so forth. But it's still a great need for other therapeutics for the brain, for aging brain. So this is a placebo-controlled, double-blinded study that was done as a collaboration with my colleagues at Washington University. And so the PRIMATE research was actually done there, and we were the partners at Vanderbilt. The way this study was done is it was a unilateral carotid injection of MPTP to lesion the substantia nigra on that that ipsilateral side. And the monkeys were then treated for 2 months after the disease had been initiated. So these were post-treatment. It was a post-treatment protocol, and the behavior, PET imaging of the dopaminergic system was carried out throughout the study, and then brain pathology was looked at the end of the study. This is a PET image showing a real loss of dopamine neurons and dopaminergic content in the brain with placebo-treated animals. And then we have real preservation of the Nigra in the UP1007 treated monkeys. This translated into behavioral benefits. And so we have a Parkinson's rating scale. It's used also in humans, and it's an 18-point scale. And what we were able to see is that there was a gradual improvement with UP1007 on this Parkinson's rating scale. And at the end of that 2-month treatment, it was quite significant behaviorally. Other parameters are not on this slide, but those also showed significant behavioral benefits in this primate model. But to allow us to translate these findings to people and to specifically be able to look for inflammation as part of this process for 2 reasons: to be able to potentially screen patients coming into a phase 2 trial for inflammation in the nervous system, to basically enrich our first population of Parkinson's patients, but also to document target engagement of our anti-inflammatory molecule. We have collaborated with WashU and with a group at UPenn to develop novel PET tracers that will detect inflammation. And this is a primate brain showing one of these PET tracers that actually is quite capable of detecting inflammation in multiple organs, but of course the target we were looking for most clearly was for brain imaging. Bringing this back to the primate model, we've been able to document that there's a delayed inflammation in the cortex above the substantia nigra, and this, we believe, may be a proxy for the inflammation that may be contributing to cognitive impairment in Parkinson's patients. So Parkinson's dementia is, of course, a very large component of disability for Parkinson's patients, and it is not well treated by any of the current therapies that do help with the motor problems. This is the tissue level of inflammation is also correlates very well with this PET documented inflammation. And so this is brain tissue from that same region showing astrogliosis, the bright green cells here, and also microglial activation as shown by enhanced microglia, and also morphology changes we know are associated with inflammation and inflammatory activation of microglia. And the animals treated with UP1007 have a normalization of this, so that there is actually very little evidence of inflammatory activation in the animals treated with UP1007. So as I mentioned, this is nearly 20 some years worth of research, and we have the NIH and the Veterans Administration and actually the Department of Defense to thank for a substantial amount of funding that has supported this research. So this is late stage research that also has been supported by certain foundations as well, to the amount of about $39,000. And so this is actually mature, to some extent, mature technology that's been supported by other funding. So I just wanted to mention a few of the areas where we actually have publications in, as I mentioned, the Parkinson's disease in primates, but we also published efficacy in an ALS model. We also have multiple publications in natural aging, and I'll show you just one slide on that. And we have very recent publication documenting reduced activation of leukocytes and preservation of blood-brain barrier in an ischemia model. I know I'm close.
One minute, Laura, please. Thank you.
Yep. This will be the last slide then. And that is simply that we did a long-term aging study, and it was not intended to be a lifespan study, but it actually was able to show improved cognition and enhanced lifespan in this long-term 300 plus mouse study. And we believe that's due to a decrease in cancer and renal fibrosis. And that actually is basically the main focus of our research and our future, plans, corporate plans. Thanks.
Excellent, Laura. Well, covering a lot of the basis there for a lot of very interesting outcomes for neurodegenerative diseases. So good luck with your work.
Thank you. And I'm sorry
Thank
I
you
ran
very
so
much for your time.
Thank
No,
you.
that's perfect. Thank you.